So today we're going to talk about lactic acid fermentation, which is an important part of our physiology, as well as being an area of active research in the field of metabolism. So let's just dive right into it. So I talked previously about cells using glycolysis to generate ATP, and a basic idea of this is you take a glucose and you break it into two pyruvate, sorry, and in doing so you can generate two ATP molecules. And this is a net favorable reaction that can be quickly run when a cell needs energy. The problem is actually this isn't a net energy generating pathway by itself, and the reason for that is we're taking NAD plus and we're making NADH as part of this. We are oxidizing the glucose as we're making pyruvate. We're sticking electrons onto an NAD. And this is just accumulating, right? This is just building up. There's nothing to prevent that from continuing to build up. And if we don't do something that, we're actually going to have to make more NAD+, right? We need to continue to make NAD+, so we can keep running our energy generating pathway. But the problem is it takes more than 2 ATP to make an NAD+, right? If you just do the work on the sort of metabolic pathways you'd need to go from to build an NAD+, it's way more than 2, which means if you run glycolysis by itself, it's actually going to be a net energy consuming pathway rather than an energy generating pathway. So how do we get around that? Well, it turns out there's a couple of strategies cells use, but one of the major ones is fermentation. And what happens in fermentation is we take pyruvate, one of the products of glycolysis, NAD+, another one of the products of glycolysis, and we do a reaction with them to generate this new molecule, lactate and NAD+. So we're recycling our NADH to NAD+. Cool, that's exactly what we need to do. The way that we're doing it is we're doing a redox reaction. We are taking a hydride off of the NAD+, we're sticking it onto carbon-2-pyruvate, generating lactate. So we are oxidizing NADH and we are reducing we are oxidizing NADH and we are reducing pyruvate to form lactate. Whew. Okay. This is a favorable reaction, and we can see that by looking at the standard state reduction potentials of this. If you want, this is a good chance to pause and actually work on this. How favorable is this reaction? So go find your pause button. All right, assume you paused. Let's just go through this. I won't go through this problem in too much detail other than to say we can know that the electrons are going to end up on the lactate because it has a higher standard state reduction potential than the NAD plus NADH reaction. So the electrons, this has a higher affinity for electrons. That means we're going to have to flip this reaction, and this is where the electrons are going to come from, and they're going to go to there. We can also just look and say, oh man, there are 2 electrons on both sides. Those are going to cancel. So that lets us get all the information we need to figure out the free energy. And it turns out we've got 2 electrons, n equals 2. We're going to multiply this by 96. And we're going to multiply that by the difference in the standard state reduction potentials with the donor first. So 185, right? It's going to end up there. I said donor. The acceptor first. So we're doing E acceptor minus E donor. So that's going to be 0.315, right? So we're doing, where are we going? We're going to 1.185 from 0.315. And if you plug in all those numbers, you get 25 kilojoules per mole. So it is favorable by 25 kilojoules per mole to do this redox reaction. And it's really important that this is favorable because we're trying to use this as part of an energy generation pathway, right? If this isn't at least neutral, then we're going to be losing energy rather than making ATP out of glycolysis. So then overall, if you look at it, the net reaction for glycolysis plus fermentation is a glucose plus 2 ADPs and two inorganic phosphates going to 2 lactates, 2 ATPs, and 2 waters. It's just a very satisfying reaction, right? It's just a very simple, straightforward. You take a carbon molecule, you get out 2 more carbon molecules, and in the process, you make some ATP. So the great thing about this reaction is it can run forever, right? As long as you have something to do with that ATP, where you're going to take the ATP and generate it to ADP, which cells do all the time, right? This can just keep running. As long as there's glucose around, you can just run this reaction. You're not accumulating anything. And so what we can see then is that this reaction is mass balanced. We're not accumulating things. And it is also overall quite favorable. So this is, if you add up all of the standard state reduction potentials for glycolysis, it's about minus 50 kilojoules per mole. We just got this from fermentation. So overall, this reaction is minus 75 kilojoules per mole favorable. So it's a really nice reaction, right? It conserves mass, it can run forever, and it can release energy. Now, the aside here, now for those of you that have thought about things like aerobic respiration, obviously it's not releasing as much energy as aerobic respiration, but it is releasing energy, and it's doing so quickly. So it's still an important part of our metabolism, even if it's not something where we're generating, all of our ATP when we run a marathon out of this. We still are using this extensively throughout that process. Okay, another cool thing about these reactions is that they are quite efficient. And one way we can see this is by looking at the energy that's stored in the bonds of the molecules. And so what I'm showing down here is glucose being completely combusted in oxygen. If you do that experiment, you light glucose on fire in the presence of pure oxygen and measure the heat that's released, it turns out it releases 2,820 kilojoules per mole, approximately. Well, not approximately, with some precision. If you do the same thing for two lactate molecules, what you get is that these things release 2,726 kilojoules per mole of two lactates. So this number is not quite as high as this number here, so it releases less energy. So there's less energy stored in the bonds of lactate. But what's really interesting is if you take the difference in these two numbers, you get a pretty small difference. So what we're going to do, we're going to borrow. I'm actually subtracting on the fly here, 7. That is an 11, and 11 minus 2 is 9. So it's 94 kilojoules per mole. That's the difference in the energy of these two molecules, the energy stored in the bonds. And ATP is about 30 kilojoules per mole to make, which means, and we got out 2 ATPs out of this reaction, which means we captured 30.5 times 2 the energy. So we got 61 kilojoules per mole of energy. That's what we captured. But we only burned 94 kilojoules per mole. So this thing is like 66% efficient. Actually, I think I have the real number here, 65%. This is an amazingly efficient engine, right? We released 94 kilojoules per mole. We captured 61 kilojoules per mole. So the whole system is an amazingly efficient energetic machine. For comparison, a gasoline motor, a really efficient one, might be 30%, right? So this is much more efficient than many machines that we build. So it's a very good way to sip every last bit of energy out of of glucose to lactate. Okay, that almost made sense. All right, so cool. I said we're making lactate. What do we do with this lactate once we generate it? And it turns out lactate and pyruvate are readily exchanged with the bloodstream. They do so through these transporters called monocarboxylate transporters, MCTs. And MCTs are basically passive. They just let molecules follow the concentration gradient. So if there's excess lactate in the blood, it'll diffuse out. If there's excess lactate in the bloodstream, it'll diffuse in. Lactate is actually it's the second highest concentration carbohydrate thing in the blood. So it's about 1 millimolar often. And glucose, by comparison, is about 5 millimolar. So it's nearly as high concentration as glucose. And pyruvate is about 0.05 millimolar. So lactate is kind of one of the bigger contributors to our overall blood chemistry as far as carbohydrates are concerned. If we are doing a lot of lactic acid fermentation, so for example, we are working out really intensely and we are not getting enough oxygen to our blood to run, or to our muscles to run aerobic respiration, what we'll actually see is lactate builds up here, right? And then remember, those MCTs are concentration gradient dependent, so we're going to start exporting lactate. It's going to start circulating in the blood. Turns out the liver can take up lactate, can run a process called gluconeogenesis with it, and then can dump that back into the bloodstream where it can be taken up and consumed by the cells. This is known as the Cori cycle, and it's one of the major fates, you could say, of lactate as it gets put into the bloodstream. It gets recycled in the liver into glucose and fed back to your muscles. But it's actually generally, we think about this as the Cori cycle has been known for a long time, and we think, oh, this is just, we got to deal with this waste product. But it turns out basically any cell that's doing aerobic respiration can actually use lactate, right? So if there's lactate in the blood, a cell can import it, can convert it to pyruvate, can then run the citric acid cycle and make a lot of ATP. And so one of the ideas right now that's being developed is that lactate's actually just a general shared pool of fuel that is used by many different cell types, right? So lactate goes into the blood, and it's just this shared resource that everybody has access to. Rather than being a waste product that has to be processed by the liver and the Cori cycle, it's just a general shared food or fuel that's processed by a few cells that are really intensely, say, running glycolysis and fermentation, and then the other cells can use that as an input for other processes. So it's a mass neutral process that releases energy and is energetically efficient. It also might generate lactate as a generic fuel that's used by many different cell types. The last thing, and this is a pretty exotic idea, but I love it so much I just have to talk about it, is that maybe lactate and pyruvate actually act as a redox buffer. It's a little bit of a complicated argument, but I'll walk through it because it's pretty nifty. The idea here is inside of our cells, we like to maintain an excess of NAD plus to NADH, right? The idea here is the excess NAD plus gives us oxidizing power. So we can do things like glycolysis where we need to do an oxidation reaction. One of the interesting things about lactate and pyruvate is in your bloodstream, it's actually not really that important what their absolute magnitudes are. I mean, it does matter some. But actually, what really matters is the ratio of lactate to pyruvate. And in fact, if you look on the Mayo Clinic's lab website, I'm going to disappear again. These slides are not well designed for me to be in front of them. Sorry. But Mayo Clinic's labs is a really interesting resource because you can basically look at their testing catalog. Say, if I'm a clinician and I want to do a blood test, what was the blood test I can run? And then what is the clinical interpretation? And one of the things you actually do when you're doing a lactate pyruvate panel in blood plasma is you look at the ratio of lactate to pyruvate, not necessarily at their absolute values. So if the ratio is weird, that's a problem. But the ratio just generally tends to be, around 20, right? So the ratio is pretty well fixed. In fact, if you look, above 20 is bad, below 10 is bad. I know it's really small. But that basically means that those numbers I gave of 1 millimolar to 0.05 millimolar, that number, that ratio moves as a fixed ratio. Okay, why does that matter? Okay, so first observation is we have a lot of blood, right? We have approximately 5 liters in a human. Cells are a lot smaller than that, so they have, 10 to the 12th liters as a guess. Lactate and pyruvate freely exchange with the bloodstream, right? So this is exchanging based on concentration gradients. They can flow back and forth across the bilayer with these NCTs. But NAD plus and NADH can't exchange. So what that means is the NAD plus and the NADH in here can respond, their ratio, we'll see in a second how this happens, their ratio can change in response to changes in the blood ratio of lactate to pyruvate. So let me show you what I mean by that. Okay, so I'm going to do a simple exercise that is going to look like a lot of math, but it's pretty simple math. I'm going to ask the question, if lactate and pyruvate equilibrate in the blood, what is the NAD plus NADH ratio in the cell reflecting that? And so we're going to use this expression up here, which is our delta G equals delta G naught prime plus RT log of Q. And we're going to say that we're at equilibrium. We're going to let the system equilibrate. But remember, we're equilibrating with the blood. So the blood is this huge thing that if a cell equilibrates, it's not going to change the concentrations of the things in the blood. Rather, it's going to change its concentrations. The lactate and pyruvate ratio is set by all of the molecules in your bloodstream. So if a single cell equilibrates with the bloodstream, it's not going to change lactate and pyruvate. It's going to change the things that it can change, which are the things that can't exchange with the environment. Okay, so let's zip through this. I'm just going to show some algebra. So get ready to pause if you want to look at the algebra. I'm just going to show it and then show the answer. But basically, I'm doing a rearrangement with some log rules to get to this expression at the bottom, which is saying that the ratio, that the NAD plus to NADH ratio in the cell is going to equal the pyruvate lactate ratio in the blood raised to the delta G naught prime. Okay? What does that mean? Well, what that means is we can plug in some numbers. So if we have a 20-to-1 lactate to pyruvate ratio in the blood, so there's a 20-fold excess of lactate in the blood, and we have a delta G naught prime of minus 25 kilojoules per mole. We calculated that earlier, right? That's the energy to do this reaction. You plug in the numbers, and you get the ratio of NAD plus to NADH in a cell should be 816 to 1. So if in fact lactate and pyruvate can exchange across the bilayer, which they can, and if they are in this massive excess in the bloodstream relative to each cell, which they are, what this is saying is they will tend to drive the NAD plus NAD ratio in the cell to be what our cells wanted anyway. The cells can basically outsource by doing fermentation and dumping lactate and pyruvate into the bloodstream. Cells can say, cool, all the cells are collaborating together to set this ratio of 10-to-1 lactate to pyruvate. Then every other cell can just plug into that, and that's by plugging into the bloodstream, they're actually setting their NAD plus NADH ratio. They're basically connecting to a source that drives this ratio so that they can do chemistry. So they're outsourcing the requirement to keep these at a particular ratio to the entire bloodstream and the entire metabolic system. Okay, that was a little bit of a wild thing. It's actually something that people are still discussing in the field right now. But I think that the idea here is this glycolysis fermentation combo might actually help maintain high NAD-NADH ratios across tissues. So, when you're doing lactic acid fermentation in your, muscle cells, it's actually helping your kidney cells maintain an NAD plus to NADH ratio that's useful. Okay, so that's all I have on lactic acid fermentation. Hope it was interesting. I look forward to diving into some more of the central carbon metabolism in some future videos.